let's start with a little bit self-advertisement. So as I mentioned, it's me, Bonidi Kumar, and then this is Francis Lopez. We are going to, help you to learn a little bit about bioinformatics for next 5 days. So we have at least 4 faculties today for next 4 days to deal with this course. Dr. Amit Rawat is here, Manpreet is here, and me, myself, and Francis. We will be also joined by Jomin Choi. He will be here on Thursday, hopefully, and he will be also leading variant analysis, that part of the course along with Francis, and he will be here on Thursday. So he's from Department of Biomedical Sciences from Korea. He used to be in Yale previously, but now he is an independent faculty in Korea. So he will be traveling all the way from Korea to join us on Thursday, and he will be there with us on Thursday and Friday. Apart from that, you will have few interesting talks to attend. Every day 5 to 6 we have a talk, which basically to give you a sense that how you can use NGS for a real life case. So we will be, so first we have Ganeshwar. Hopefully today I am just still waiting for his response. If I do not hear from him, and unfortunately I did not realize that today is Chhat. So those people who are from Bihar and UP, and he is from Banaras, so from Gorakhpur. So essentially he might be within that Chhat thing, and I could not hear from him yet. So if that is the case, then probably I will be talking, if time allows, then I will be talking about pediatrics, using NGS for pediatrics and newborn screening, but otherwise Ganeshwar will be here to give a talk. Our director for bioinformatics at Yale Center for Genome Analysis, James Knight. Jim will be talking more about best practices in bioinformatics and some of those aspects of data analysis. Then we have a clinician investigator, Rick Pierce, who will be talking about MODS and how the actually multi-organ dysfunction in neonatal ICU kids, and how he is using genomics to study those kids, and especially the origin of MODS in those kids. Then we have Madhav Menon. He was an alumni of this place, PGI Chandigarh. He did his MD and residency from PGI Chandigarh. Currently, he is also director for kidney transplant center in Yale, and he will be talking about how he is using NGS based analysis for mismatched loci to identify whether transplant rejection is happening, and you can see some of those analysis where you can figure out if those rejections at very early stages, even the stage when you cannot detect many antibody related to some of those current practices that we are doing. So he'll be talking more about that, and of course Jomin Choi, he himself will be giving a talk on how he is using NGS to study rare diseases. And so you will have a series of talks you will be hearing where how the NGS is being used. Then this course is actually, there are more finer points to this schedule, but this is a kind of a broad outline. We will be introducing today you with some basic bioinformatics tool, and then from there we will be going to experimental design and transcriptome analysis tomorrow. We'll consolidate on that theme on Wednesday with bulk RNA-seq. So you will see some of the basic thing on Tuesday, and then we'll consolidate that idea more into bulk RNA-seq on Wednesday. And Thursday will be going one more step further, and we are actually going to learn single cell RNA sequencing. And finally on Friday we'll be doing variant calling and interpretation of structural variants, single cell SNPs and all. So as you see, this workshop is actually structured in a way. It's a progressively increasing difficulty format. So, and then you need to be here every day to learn if you want to make use of it completely. So who we are? So me and Francis, both of us are from Yale University, and if you don't know Yale is actually in top 10 universities in the world and second ranking university in US. And so I know ranking doesn't mean anything, but still just to mention that, I say to put things in perspective, that's why I just mentioned that. So these universities, Yale University along with Harvard, Princeton, Brown, they all comes under a group of universities called as Ivy League. Ivy League, you may think it's because people are envious about it, but no, that has nothing to do with that. It's just because that was a sports league which they were part of, but they are the oldest school in US. and they are one of the best schools that US offers as a part of higher education. This is the second oldest university in United States. It's almost 250 years. Second or third? Which was the second then? Harvard then?
Harvard is the second.
What was the first?
It's a college called John Mary something.
Okay. So I'm correcting. So it's a third. So they started. So the history is they started Harvard for imparting Christian education when Britishers came to US were colonized, and at some point they realized that Harvard is becoming way more liberal than they wanted it to be. So they established Yale to again emphasize on the Christian education. But unfortunately, Yale became more liberal than Harvard, and it is one of the most liberal-leaning universities in the world at this point. Okay, there is a connection to India with Yale University, and it's not a good connection in a way. So when the initial building of Yale was being conceived, and they were looking for funding, this funding came from a person who was part of East India Company and posted in Madras. So he took bales of cotton from there and sold it in European market, and that money was the basis of the first building of Yale. Second thing, and then he actually collected a lot of riches from exploiting the local people, and that was also actually became part of Yale's some of the initial funding. And so recently there was Yale actually instituted a project where they were studying about slavery and how the slavery, how actually Yale and slavery was actually interacting to each other. And one of the things they apologized to, after the project was completed, they published a book about it, and they apologized to Indians and as well as some of the other people who were part of those oppressive part of. But it was not like Yale did something, but it was some of the money that came from such a background, which at that time it was not. So this is the thing. And now also Yale offers the advanced training for Indian administrative service professionals. So those who pass IAS and complete everything, and after 5-6 years of their work, like their job, they go for advanced training, and one of those trainings are conducted in Yale still now. So they almost, it's I think 8 weeks training that happens in Yale. So so Yale is that way. It's interesting. So we are located in a place called as New Haven. New Haven is almost 1.5 hours from New York and 2.5 hours from Boston. So we are between New York and Boston. So those are the references probably you are familiar with. So it's an old town. It's old. Okay, so Yale has a big endowment, like it's a private university, so a lot of money we get. We have current endowment level is approximately $42 billion that Yale has, and then we have around 6,800 undergrads. There are 8,750 postgrads and around 6,000 academic staff. So, and Yale is actually divided into 13 schools, including School of Medicine is one of them. And Yale's law school is considered as one of the top in the world even. So there are 2 law schools which are competing for this honor. One is Harvard and second is Yale. The difference is Harvard people go for corporate laws, while Yale people are more idealistic and they go for Supreme Court clerkship and those kind of thing, and pro bono, non-profit. So a lot of idealism is there in the Yale law school. One of the interesting thing about Yale is that Yale gave almost 7 presidents to United States, and this administration, interestingly, the vice president and the second lady, which is vice president's wife, Usha Vance, both of them are Yale alumni, and they did their law school from Yale. So, and you know that Usha is from India, so again that is something. And then famous Indiana Jones movie, Kingdom of Crystal Skull. This was shot in Yale. So those who are fan of Indiana Jones, genre of movie. So it was third movie, the Indiana Jones.
Third or fourth.
Yeah, one of those. So that was actually shot in Yale. So you will see that. Now coming to the Yale Center for Genome Analysis, which I am the director for operations, as well as Francis is there as a bioinformatics associate director. We are located in West Campus. So Yale has a main campus and also a West Campus, which is almost 8 miles. 8 miles means 13 kilometers away from the main campus. We have shuttle actually frequenting between these two campuses. Every 15 minutes we have shuttle to go from one place this to other. So we have a dedicated around 7,000 square feet of sequencing facility, and as we said, that this is the West Campus. This is how the West Campus, Yale West Campus looks like from the aerial view. We are located in, this is just the geographical location. We are located in New Haven, and this is New York actually, Long Island, and we are here. So this is just to orient you with the geography where we are with. So as we said, like we have 7,000 laboratory space, and actually we have another 4,000 square feet area which is for office and other space. So we have total 11,000 square feet of area. We have 35 people working with 8 PhDs and a lot of masters and bachelor's people there. Our data, whatever we are producing, that is live streamed to a high performance computing system, and this computing system consists of around 400 nodes and 4,500 or 4,500 cores, and each of those cores have 32 gig RAM and 16 GPUs are also there. So, but we are not using much of those GPUs for our work yet. But and then we also have 2 Dragon servers which is GPU accelerated, and we are yet to use that. Again, the older version of our HPC was cooled by like the what they call door cooled. They were door cooled ones. So we were spending a lot of money for electricity bill. Almost our per month electricity bill was $100,000, slightly over $100,000 just to pay for electricity for cooling the facility. So for the new HPC when we bought, we moved to a water cooled processors, which means like the water will flow through the processor and it will take out the heat. So that means we reduce some of the cost of our electricity bill through that. Now we are moving to a larger center, which a collaborative effort between Yale, Harvard, Tufts, Boston University, I think MIT, and one other. So it's a collaborative center of data center we are creating, which is called Mass Green, and it's in Massachusetts, and we are moving all our infrastructure to there. But the key point is that we have a 10 gig optic fiber which transmit data from our sequencing center to directly on a real time basis to the data centers, so that data is stored and processed there. We offer a variety of services from our facility, and this is kind of a laundry list of that, and you can see anything that you can think of we are doing. Apart from this, we are also doing a lot of clinical services. We are doing rapid whole genome sequencing. Rapid whole genome sequencing, if you are not aware, it is for NICU PQ kids, those who are severely ill NICU kids. We sequence data and report our analysis within 30 hours. So that means from the blood collection to data generation and reporting, it is done by 30 hours, within 30 hours, and that report goes back to clinician for clinical interpretation or clinical action, not interpretation, clinical action. We have a pretty good solve rate. We are almost at 50% solve rate, which we are really proud of. Probably those kids are really sick, so we usually get a reason why they are sick. So, and this is one of the fulfilling part that we do those kind of operations. So other than that, we also do tumor profiling. So hematological tumor profiling we do from our center. We also do profiling for neurological tumors like glioblastomas and meningiomas and all. And then also we are now currently working on solid tumor profiling with the FFP sections. We are just waiting for the final regulatory approval, but we have done most of the groundwork for us. So once it is done, we'll do that. Other than that, we do analysis for newborn screening and newborn kids with dry blood spot. So a lot of these things what you are seeing with the dry blood spot based is for the newborn screening programs. So we support some of those newborn screening programs from YCGA, and we use all this exome and whole genome sequencing from blood. So I don't know much about the regulations in India, but in US you cannot take, like, unless there's a reason to draw blood from a newborn baby, you only can take like a prick, a heel prick, and you can take only 2 prick cards. So that is the only you are allowed with. So that means you are working with a scarce material and you have not much to do. So we have done a lot of technological innovations on that side from the extraction of DNA to library prep and sequencing and everything. So last 2-3 years we have done a lot of those things, and then we are now at a stage when we can do these things. Okay. So history. So it all started as a KEC. We got a grant in 2008, and Dr. Shrikant Mane and Dr. Rick Lipton. Rick is currently the president for the Rockefeller Foundation. He was the person who first discovered hypertension gene, a very accomplished scientist. He was chair of genetics department at Yale and currently president of Rockefeller. So they both started with the NIH grant in 2008, and so we started a YCGA, and Yale actually provided us with a $10 million grant for establishing this. So which we, and still we have that money because we spend that money on instruments and then we recover the cost through our services. So we still have most part of that money that Yale gave us. Then the first NGS paper on clinical diagnosis came in 2009. This was the science paper, and this was the beginning of precision medicine. So first report of a precision medicine where they have correlated a gene and with a clinical diagnosis was this paper, and this came from our lab. And we were also instrumental in developing exome sequencing. So a lot of those, and Francis was one of those earliest people who analyzed those data, I believe. Is that right? Yeah. So these were the people who initially start. So we are doing microarray, and then they use that solid platform and took into a liquid phase, and they hybridization based analysis. They actually established those protocol, and Francis and his group were at that time doing the basic analysis. So they established exome sequencing. So you can see some of those early remnants of historical people who are involved in developing exome sequencing. So and we were proud to be part of it. So 2010 we bought our first Illumina machine, which was a GA2X machine, and it became operational, and we established a CLIA. CLIA is basically a certification that you need to, if you are running a clinical sample and you want to report it. So if you want to do and this is given by a College of American Pathologists, so which is CAP, stands for that, and this certification you have to maintain. Every alternate year you have to undergo a certification process, and every alternate year you have to also go through state health department recertification. So this is a very extensive, laborious administrative process, and unfortunately I have to do that every year after year. And you can see like this much area, like almost these 2 rows of files are there. Still they want papers, like you have to print it and keep it. They won't allow, they don't want any and in fact actually last COVID time when they were doing this CAP inspection, they told us that okay, you can submit some of those things online, and that was more complicated than me giving it the paper one. So and we have to maintain all those, like since 2010 all these papers are there and you have to maintain it. So it's a process. So we got our certification, and then and we have to keep validating if we are doing any new assay for patient care. Okay, and then our first bioinformatics group was started in 2012, and we got our first Illumina sequencing, HiSeq 2005, a second Illumina machine, which was the latest at that time was 2500, and then 2015 we got our next Illumina machine 4000, that was a new one, and then 2017 we got 6000, Illumina 6000, and 2017 itself we got our sequencing, a 10x machine for single cell RNA sequencing, and 2022 was another year when we just came out of our COVID, and then we established spatial technologies, and I don't know how many of you are aware of this. So this is a kind of a technology where you analyze single cell on a tissue context. So you will be also looking at the position of cell, and then you have a single cell transcript analysis. So we have 4 different spatial platforms which we established in 2022, and in 2023 we purchased our most latest state of art NovoSeq X Plus and a NextSeq 2000 at that point. And so we have, so this is kind of our history, how we published more than 1500 papers, and then many of them are in Nature Cell Science, and we attracted almost more than $500 million in grants and funding. So, and we actively write grants. We also support other people to write grants for genomics space. The one of the thing that I don't know whether you you could perceive it from the presentation, from the history, that every time a new technology is introduced, YCGA is at the forefront of acquiring it and providing that to the researchers. Because unless you give the state of art technology, you cannot leverage the cost and other things, that advantage that comes with that advancement. So from the 12 GA2X sequencer, which was the beginning of YCGA, now we have 2 NovoSeq X Plus. So NovoSeq X Plus is a state of art Illumina sequencer at this point. We almost generate around 7.5 terabases of, 15 terabases of data in a 48 hours run, which is actually, if I want to convert this into billion bases, we generate 52 billion reads every 2 days from YCGA, and we are generating around 1 petabases of data every year, from 1.1 petabases of data from YCGA. Approximately we are running 30 to 36 flow cells a month, which is like around, say, somewhere between 6 to 8 flow cells a week. So this is the scale of operations that we are doing, and because of that we are able to achieve scale of economy. And our current sequencing cost is $1.05 for 1 million reads. So I need to put that also in perspective. One burger, so don't consider McDonald's of India. The McDonald's of US is the cheapest food that you get in US. So the cheapest low income people's food, you can say, is actually cost $6 for the cheapest burger in McDonald's at this point. And so you are getting a million reads for 5 times cheaper than a McDonald's burger. So this is the kind of scale of economy that you can achieve through having the new technology and the throughput that we can think of. So this also created a situation where people started moving away from exome sequencing, and now they are doing whole genome sequencing, because now the cost of exome sequencing and whole genome sequencing are almost comparable, like a few $100 more than, which means like you have more data, and then you are, so we are seeing that conversion, and we are also currently working on whole genome sequencing for clinical applications, and we are doing some pilot studies on that. So as I mentioned, all these technological innovations led to a decrease in cost, and we are actually capturing this decrease in cost by introducing new technologies and new sequencing platforms at YCGA. So YCGA has multiple sequencing platforms at this point. We have one MySeq, we have one NextSeq, we have 2 NovoSeqs, we have one Element AVITI. By the way, actually I wanted to mention, Dr. Rawat, that this is a platform that probably you should look into, okay, instead of your NextSeq 2000. Element AVITI, the cost of sequencing is almost equal to the 6000, NovoSeq 6000. So you will get a medium throughput sequencer at the sequencing cost of a high throughput sequencer. So this is a very good platform to do. Then nanopore MinION, we have a PacBio Ravio. Ravio is the latest PacBio machine, and we also have a PacBio Sequel 2. Yeah. I was mentioning about spatial technology. We have at least 5 different technologies, and we will be seeing some of this when I'm going to teach you more about the next gen sequencing technologies in 2 days, different days. One I think today, and then other is on Thursday, I believe. Okay, so we have Visium, we have Curio, we have Atlexomics, we have Cosmex, and we have Xenium. So we have 5 different platforms for spatial technologies at YCGA. And with that, I would like Francisco to talk more about the workshop.
Well, you already heard about the workshop a little bit while we were waiting, right? So what is the workshop, right? We want to be a hands-on workshop. So we want you guys to be able to do some analysis or use the tools, etc., right? But you also notice, I noticed in the audience there were a few people mentioning single-cell RNA-seq, other people even mentioned epigenomics or things like that, right? So the field is so vast that we are not able to cover everything, right? We are not going to be able to cover every single analysis. Even if we talk about single-cell RNA-seq, we could have a single workshop about it, right? So this is not about it. So we are not able, we are not going to be able to see everything, right? One of the ideas that we have about, okay, what topics we're going to choose for the workshop was about, okay, what are the things we do the most at YCGA? So the things we're going to see like bulk RNA-seq, single cell, and variant calling applications are probably the three most applications we do at YCGA, right? I would say, I don't know, Bonnie probably know better than me, but I would say at least 80% of those falls into those three categories, right? Single cell is, of course, is now the new fashion, and probably we do a lot of it. But bulk RNA-seq, we still do a lot of it, right? Maybe not at the cost level, because it's really cheap to do bulk RNA-seq compared with single cell, but at the number of samples, I wouldn't be surprised that it's still quite common. And we still do a lot of whole exome sequencing and whole genome sequencing. So that's the reason we pick those three topics. And this is also really important, right? The idea of if we want to be a useful workshop, right, we need to pick things that not only about the technologies or the analysis types, we want to teach you something that you can take from these and use it in something else, right? So we talk about epigenomics, I think somebody mentioned it, right? So if you can, there are a lot of commonalities in all the analysis we do that you can take them and use it somewhere else. It could be using R. You can use R not only for single cell, you can use it for bulk and other type of analysis, or even just producing plots for your papers. Or if we talk about Jupyter notebooks, you're going to be able to use them for anything you do, right? Probably in the lab regarding analysis. And within the analysis types, right? So aligning sequences to a genome is something you do for a lot of things, not only for bulk or variant calling. You're going to do it for single cell or even methylation analysis or anything you do. There's a lot of commonalities in all the types of analysis. We give you a little survey before the workshop, right? So this is where the answers about the knowledge of R. So this is a big challenge, right? So the people that comes to workshops, and this is common, right? We see this all the time, that when we organize a workshop, you're going to have all kind of levels of knowledge, a prior knowledge before coming. So, and it's complicated to prepare a workshop where you're going to, make everybody happy. So if you do it too complicated, then the people that's new to the field, they're not going to learn much. If you do it too easy, then these people get bored. So one thing that I recommend is we're going to do exercises in groups, right? So, and individually, both. But, don't hesitate to help somebody. So if you fall in the side of, oh, I already know how to do things in art, and you see somebody struggling, please help him. So I think in that way, so if you already know the topic, you can help somebody else and maybe learn something else too. If we talk about, so the goals of the workshop, right? So one of the things, you're going to hear talks, right? With those talks, we will offer, so the type of resources that you have to do with the NGS, right? It's not only about sequencing, there's all applications of that NGS. So the talks are going to give you some ideas about the different applications we use NGS for. One thing that I noticed, I think it's important, and maybe it's boring for everybody, but I think it's important to understand the data formats that we use when we do data analysis, right? I know this is not interesting. Sometimes the only thing you want is to run a pipeline, get your results, and that's it. But there's a lot of troubleshooting when we work in data analysis, a lot. And part of it, you need to understand the type of data formats that we use. What we said before, right? We're going to try to give you tools that you can make use not only for the things that I mentioned, the data types that we're going to be working, so you can extrapolate to anything you do about data analysis. Well, of course, that's the main idea. A lot of people come here, right? Half of the people come with the idea, I'm going to be able to do my own analysis. Well, I don't know how much you're going to learn here, but I hope you'll learn quite a bit, at least to get your feet wet on the field and learn more things in your own. And this is something that I always mention. So I'm part of the offering service most of the time, right? We help people to do data analysis, or we collaborate with people, with investigators that they only work in the lab. And there's always a barrier of communication. So one thing that I always say is invite people that only work in the lab is to learn this, right? How to communicate with a bioinformatician. If you know the language, if you know how to communicate, so both sides of the equation, they're going to learn a lot of it, right? They're going to be able to produce better data and better analysis. We're going to talk the first tool we're going to be using. It's not related to data analysis, but this is a website. Are you familiar with Poll Everywhere? Do you use it in classes? Oh, you use it sometimes? Okay, so if you don't mind, go to this website. So, and if you can, you should bookmark it because we're going to be using it through the talks maybe, and at some point we're going to be using this website. Or you can use even your cell phone, by the way, for this. You don't have to use a laptop, but if you go to the website, keep that tab open if you can, in your phone or your laptop. I don't think it needs to be uppercase. I did it uppercase so you can see there's always confusion about the L, if it's an I or not, if it's lowercase. Yeah, everybody is in the website. So why we use this? So
We will need the password for the Wi-Fi.
oh no, there's no password for this. There's not going to be password. Don't worry about it. No.
If they want to access this, they want.
Oh, for the wireless. Oh, okay. So it's immunology 5G, and the password you know better than me. I forgot.
So the immunology 5G and the password is LOGY, all small case. LOGY.
Oh well, I shouldn't
Go to
put it.
check.
Oh, I don't know. Yeah, you can. So
Can you open it?
So if you want to type it, you want to type it here.
Yeah. So both of them will work. There is one called immunology 5G and the other one is 4G, I guess. 2.4. So we can connect either. If you are able to connect 5G, it's better. The password is same.
So everybody has wireless. You can say no if you don't. Good. Should we move on? Did you access the website? It's going to ask you for a username. I think you can put whatever you want or ignore it. Any questions we ask are going to be completely anonymous. So can we test this? So we're going to ask questions like this during the workshop. But I was asking Bonnie what controversial question could I ask, and say, talk about cricket, and say, I don't know. I don't know who these people are, so you can say whatever. I'm going to believe whatever your answer give me. So try to answer the question. It's not updating?
It's not updating.
No? Huh. Oh, I see it. The funny thing is I see it in my computer, but not here. Okay, let me try to go back. Why it's not? Well, I can tell you, Sachin Tendulkar is winning with 36%. Is he the best? Is
Why is
it,
it not showing there?
I don't know. I'll check it, but it's not. It's showing in my computer, but not on the screen. That's a funny thing. Probably if I do this, let's see.
Maybe you can refresh it.
I'm trying, but I don't know why it's not.
Are they able to use it now?
Why it's not? We'll figure it out. I'm going to ask you one more question, if you don't mind. Is if you perform, we sent an email about pre-workshop materials. So how many people did perform the pre-workshop materials? What? No. Oh, I'll see it on the screen.
You can actually register there in your code. You can see that on your screen.
I can see it in my screen. I don't know. We'll do a test later. Well, 42% said yes. 25 says partially. 32% no. So it's better you don't raise your hand. Oh, they're honest, yeah. And 1%, 4% is already familiar. That's probably one person in the room. Okay, thank you for being sincere, at least. But that's what we said, right? So it's going to always be a challenge when we try to do bioinformatics. And for bioinformatics, you need computational skills. So it's good. I mean, for the people who did the pre-material workshop, that's good, because you're going to learn more through the workshop if you already know what we're talking about. The way we build it, or we try to build it in a way that is going to be incremental difficulty. Well, that's the idea. Today, we're going to be reviewing part of the material that you have in those tutorials we're going to be with today. But after today, we're going to start assuming that you already know. So we'll see what happens. I think it's your turn, right? I hope I'm not carrying the microphone everywhere. Yeah.
Is this okay?
Yeah, I think it memorized whatever we tried.
Okay, that's fine.
So.
exome or a genome or a targeted panel to find the cause of that disease, which could be a SNV in the genome of that individual. It could be a heterozygous or homozygous depending upon the mode of inheritance. And then from a research point of view, you could look at mechanisms of disease. Common diseases may also have some gene predispositions which you can discover by genomics. You can look for biomarkers for different diseases, that also you could do by genomics, and then you could amalgamate all that information to do something which is known as synthetic biology. But there are challenges in these omics as well. For example, there could be missing data in different levels, or there could be no data at all. So the solutions could be that you could discard those patients where the data is missing, or you could impute data from different omics layer to cover up for that data, but it may cause data distortion. For example, if you have mRNA data, you could impute that data to think about protein expression, or you and so these are the challenges in multi-omics data. And this is just a timeline. I found this very interesting, of the timeline of different omics platforms. So I think Bonnie would be also talking about a bit of this history in his talk. So I'll just leave that. So the timeline for genomics, it started with the discovery of the DNA double helix in 1950s by Watson and Crick. Then the genetic code was deciphered. Sanger sequencing came in the late 60s and 70s. It was initially with radionucleotides, and then later it was automated with fluorescence. Then the PCR was developed by Carey Mullis, and then you had the genome project, which was a very ambitious project which was started and which was completed. And then you had next generation sequencing in the late 2000, and now you have got different platforms which are doing NGS. In epigenomics, you have the first discovery of histone methylation, then histone acetylation, DNA methylation, and so on. In transcriptomics, the initial part was with microarray, then you had RNA-seq, single-cell RNA-seq, and now you're doing also spatial RNA-seq, which I think Bonnie would be covering. And in proteomics, MS and admin sequencing of proteins were the first technology, but now you've got high throughput platforms where you can do much more. And metabolomics also, you have got LCMS or MSMS based platforms. And lipidomics also, you've got lipid maps or other techniques. And then for all this data, you've got different tools for analysis. So with that, I think I would end here, and I'd like to think that it's like an elephant, and when you're looking at one particular omics technology, you're looking at one part of the elephant, but I think it's the whole which is more important. So I think next Bonnie would be talking about the basics of NGS and its applications. Over to you, Bonnie.
Quick question. I have a question for you.
Yeah.
Two options.
Yeah.
You can actually finish talks like till 12 and then go for lunch. And then
No, lunch is from 1 to 2.
1 to 2. So,
Yeah.
so we'll finish talk at 12 and then we have 1 hour
For the next
for
thing.
the next
Yeah.
thing, and then we'll go for lunch and then come back. Fine, fine, thanks. So I just wanted to know what is the hard stop there.
So it's 1, not
1.
12.
No, so 12 I will stop and then Francis will. Okay. So, great. So I'm going to talk for, actually this is a deck of slides, so don't worry about how many slides are there. I'm not going to really, maybe I will try to go through all the slides, but I'm not going to do that today, okay. It will be done over many days during the workshop. So the idea that we should start thinking about is like what is actually genomics information or information encoded in the genome gives you. So if you think about human to human variation, almost 99.9% of humans are similar in terms of their genome sequences. And interestingly, each of us has millions of SNPs also. So which we don't know what those changes means. Okay. So, each of one are unique at one time, at the same time we are very similar also. So, which makes it very challenging for us to really make sense out of whatever variability what we are seeing. Okay. So, if you think what makes us us or what makes me is probably the 0.1% of the genome that actually makes me. And this really attribute it's into my color, hair color, eye color, height, everything else that actually comes with that. And this also related to drug responses. So, a lot of this differences what we see in human being and how they respond to drug, that actually also has to do with the small 1% variations that we have. Now, we cannot attribute everything to genes because it's too much. We can't blame or we can't give credit for anything good or bad happening to genes alone. It's actually gene and environment, that interaction, that gives you the final output what we end up being. And one example is this two fellows. One is Jim Fixx and other is Winston Churchill. Jim Fixx was an athlete. He used to train. He was a marathon runner. He used to train every day. Or you think about like marathon or runners, they have to have balanced diet and they will be eating all right thing, doing right stuff. But unfortunately, he died at age of 52. And Winston Churchill, he was a chronic drinker. He actually was a connoisseur of scotch and used to smoke a lot. And he ended up living for 90 years. So, which means that it's not just about your environment in which you are in, like you are training or something, but again the importance of genome in this process. What is the predisposition for you as a person, and when that predisposition interact with the environment means your lifestyle, your diet, your stress level, your general well-being. So, when that interact with the genomic information, but the genome gives the blueprint of that interaction. Somebody without a predisposition may not end up having, like, so you saw a lot of people actually talk about smoking. So, I can tell you one small story about my own father. My father started smoking at age of 17 and very recently he died. He died actually last year, and he died after he was diagnosed with COPD. But interesting story is this, which is part of the story is, he stopped smoking at age of 65, and that happened because my sister's son was born. He had a cardiac issue, so he stopped smoking for him, saying like he should not be exposed to smoke. So he stopped smoking for at 68, and after I think 3 years after that he was diagnosed with COPD, and he was in a ICU for some time, and then he was discharged. He came to the ward, and doctor came and asked, oh, you were almost about to die, and miraculously, he was miraculously saved. He was almost gone kind of. So the first, and then he said, did you smoke? So he answered, yeah, I started smoking from age of 17. And so he said, oh, okay. So doctor was actually not expecting the next sentence, and he said, everything was fine till I actually stopped smoking. So sometimes it's not. So this predisposition to a particular lifestyle issue is often important when we are thinking about the genetic component underlying those predispositions. So it's not just about environment; it's about the interaction of this environment. And so to understand this more, we need to sequence DNA. And so what we are going to see today in some of these introductory talks, that we are going to look at history of DNA and sequencing. Then we also will.
We are not able to see the slides in the front. We have this in the side or something. You don't have to see it.
But you can say it here.
Oh, I don't know. I can talk from here. That's not a problem. So yeah, we will be actually talking about applications. What are the applications of next-gen sequencing? Then we will also touch a little bit about pharmacogenomics and the variants, how we are actually dealing with those things. And then finally, we will be talking about single-cell sequencing, spatial analysis. Okay. So history of DNA actually don't start like in any recent time. Okay. It started way back in 1869 when the DNA was discovered, and people don't know that what is the nature of this DNA is. 1909, it was characterized. The chemical nature of DNA was characterized in 1909, and then a lot of things happened in between. People were trying to figure out that if DNA or RNA, what is the hereditary material. All these experiments were going on, and in 1953 comes the ability to understand that what are the real structure of this DNA. So, the double helix structure of DNA that was given by Watson and Crick was proposed in 1953, and that was the biggest breakthrough in. And what actually it gave you, and the significance of this discovery was two. One, we understood the very chemical nature of DNA and its structure. Also, we got ability to manipulate DNA. So, the outcome of this discovery was, which later became recombinant DNA technology, where you can cut and chop DNA and make a new hybrid molecule. You can do so many things with that. And what also it led to, it actually converted biology from a descriptional science to an experimental science. That means you now can actually manipulate the biological system, and you can measure the outcome of that in terms of system, like what is happening to those manipulation, what is the effect of those manipulation on at the system level. You can do those measurements. And then came 1977 when Sanger sequencing was discovered. And so at the same time, there was another platform or other way of sequencing came, which is called as Maxim and Gilbert. And the reason why Maxim and Gilbert was not that popular, or also not now also popular, because it was not automation friendly. So, Sanger sequencing was automation friendly. So, the first genome was sequenced, it's a bacteriophage genome, which was just 5 kb big, using Sanger sequencing in 1977. And first automated Sanger sequencing machine came in 1986. Interestingly, after all these advancements, still these machines are actually operational in labs, including one in Yale. So, people are still doing for low throughput sequencing, people are using Sanger sequencing as a means to sequence. Okay. And in 1990, people just thought that, okay, let's sequence genome. That was the beginning of Human Genome Project. So, it was not like people just started sequencing human genome, because it's a big genome. People were sequencing 5 kb, 10 kb genomes, but they were not used to sequence a 3.5 gig genomes. So, a lot of background work needs to be done before really they start sequencing human genome. So, first what they did, they started in Baylor College of Medicine, they first started a sequencing factory, and they sequenced a bigger genome, which was a bacterial genome for influenza, which was around 1.8 MB long. So, that is when they established all the ground rules for aligning, stitching, and back library preparation. And 1998, they sequenced first animal genome, which was a nematode from C. elegans. So, it's a nematode, and its genome size was 97 MB. So, now you are seeing the first one was 5 kb, then they sequenced 1.9 MB, then they sequenced a 97 MB big genome. From there, they started working on human genome, and in 2003, they completed human genome, which was about 3 GB of sequence. So, it's a big, huge genome, and it took almost 13 years and $2.3 billion. Okay. Now, I have to give you a perspective. I don't know how many of you are familiar of billion word. So, million is 10 lakh, okay, and billion is 1,000 lakh. So, that much dollar, and dollar is around 83 rupees, and now it's actually 86 rupees per dollar. So, that is a huge amount. So, people have spent that much. Then in 2000, so one of the outcome of human genome sequence was not just about sequencing human, but it also bring in new technologies for sequencing. And the result of those new advancement was the development of this new technological breakthroughs called as next generation sequencing. It is just because they were using Sangers and they wanted to give a name, so they gave it next generation sequencing. So, this is an umbrella term that is used, but the whole important aspect here is that one side they could actually sequence genome, but they also developed new ways of sequencing, cheaper way of sequencing genome. So, please be keeping this in mind, 13 years and 2.7 billion, because we will be visiting this number shortly. So, at this point what is really happening is that a large number of human genomes are being sequenced. In Sanger's Institute, they just completed exome sequencing of 243,633 genomes. All of Us program, which is a US program, they just completed 250,000 genomes, and their target is to sequence 1 million genome. UK Biobank has another 500,000 genomes, and they just released 450,000 genomes, whole genome sequencing. So, all these are exome sequences, but the UK just released their whole genome data set, which is around 450,000, like 450,000 genomes. So, lot of high throughput sequencing projects are going on all around the world, and UK also just launched, they called England newborn screening or something, which basically idea is that every single newborn baby in UK will be sequenced, and their data will go into their clinical records, and clinicians will use that for their future. Anytime they want to use that information for any kind of interpretation, they will be using that. So, this is going on, and we are also currently working on a pilot project where in US we are doing this similar newborn screening program in a primary healthcare setup. So, we haven't got grants yet, but we just wrote that a grant proposal for them. So, it's not just human, because this planet belongs to other animals too. So, a large amount of animals from every single phylum is being sequenced now. So, a large scale plant and various animals are being sequenced at this point of time, and people are using sequencing as a tool to study various aspects of biology. So, one of the key advancements that is happening at this point is that the sequencing, there is a war going between the companies for the sequencing companies to slash down the cost of sequencing, and that leads to various platforms which gives you a very cheap sequencing. And at this point there is a platform called as Ultima. They just released $80 genome. That means you can do less than $100 genome, whole genome for human. And so which is like unheard of. So the number that we were talking about, you remember, $2.3 billion, coming from $2.3 billion, now we are talking about $100 genome. So $100 genome, even if you think from Indian context, if 8,000 rupees you have to pay for a clinical test, everybody will be ready to pay for that. Is that right? So which means now, because of this cost of sequencing, the sequencing is now possible under the patient care setup. Because if you have to pay, like say $1,000 for a genome, not many insurance people will be ready to pay that. And if you have to shell out that kind of money from your pocket, it's a big amount. But $100, yes, I'm happy to pay for, $100 for that. So the whole idea is to give a $100 genome in a clinical setting, so that people can use, more and more clinicians, more and more people can use this genome sequencing as a means for various kind of diagnostic purposes. Okay, so this is really getting real nowadays. So cost of sequencing, as I mentioned, that this is dropping down significantly, and most of this is happening because of the invention of new sequences, and new companies are actually doing a different ways of sequencing it, and we will see in coming slides like how various companies and various platforms are using sequencing in different ways. So this is actually crashing the cost. Meanwhile, what we are at this point is around $350 genome for a 30x whole genome sequence for $350. So this is the price that we are charging. So compared to $2.3 billion, and this happens within 48 hours. That is another thing. And very recently, I am also going to talk about that sequencer, which is released by Roche, and they have shown that they can create a whole genome sequence in 4.5 hours. So not just whole genome sequence with a clinical report can be generated in 4 hours and 30 minutes, and this is a world record now. So they just got the Guinness. And last week I was in Boston ASHG, and where they released that information. So we are actually talking not just about the cost here. So $350 or $400 genome we are talking about, but at the same time we are able to generate data in 4.5 hours. And I mentioned about rapid whole genome sequencing, which we are generating data to sequence sequence to data within 30 hours. So which means a NICU PICU kid who is waiting for a diagnosis will be immensely benefited if you have a high speed sequencing platforms and at a reasonable cost. So cost is also, because if you are spending $1 million to gain something, it's not useful. But then with such a cost, it's actually very much useful. So sequencing history is divided into generations. So we talk about, the first generation sequencers is essentially Sanger sequencing. So the first generation sequencing is based on, there were 4 different innovations were there in the first generation sequencing. The first was chain termination method, which was Sanger's method, which was later on automated. Then you have chain degradation method, which was Maxim and Gilbert method. Unfortunately, this was a very robust method, but it's hard to automate, so it could not get much traction. Then there are two other methods. One is called as pyrophosphate sequencing, and second is ligation enzyme method. So all these four methods were developed during first generation sequencing, but the last two, pyrophosphate sequencing and ligation methods, were not commercialized till second generation of sequencers. So only the first generation sequencers were using chain termination methods. Okay, and one of the major advantage of first generation sequencers were that its accuracy. It was highly accurate. Highly accurate means like highly accurate. Very rarely you have a chance like you will get a wrong base calls on them. But the problem is you are sequencing one molecule at a time. So cost of sequencing is very high and throughput is very low. And one of the example for, as I mentioned, for first generation sequencing is Sanger sequencing. So here we are actually what we are doing something called as chain termination methods. You will mix normal NTPs along with the D-deoxy NTP. So whenever a D-deoxy NTPs are incorporated, it will terminate the chain. Okay, and each of the D-deoxy NTPs are labeled with one unique color fluorophore. So that means you will generate a series of fragments of various size wherever the termination is happening, and then when you run an electrophoresis gel, and then you can read the color from top to bottom, and you can deconvolute the sequence, the order of those sequences. So because each of the fragments are actually stacking at different level, and these colors are read through a laser. So basically electrophoresis is happening, laser is at the bottom, and it will, for the first base will pass, which is the shortest corresponding to shortest fragment. So that will be the first base. So similarly laser will read that, and the second base will pass, third will pass, fourth will pass, like that will pass. It will create something called as chromatogram. So this is how actually it looks like. The chromatogram, you can actually deconvolute this into a sequence, and the chromatograms can be converted to sequence. So this is what actually, which is what done in for the first generation sequencing using Sanger sequencing. Okay. Now as I mentioned, like once they have done with the Sanger sequencing, now it comes next generation sequencing. It just means like you created a next generation sequencer. So next generation sequencing can be divided into basically 3 different generations, and I just mentioned 2 here because of one particular reason. I will tell you why. So the second generation sequencing and the third generation sequencers. Second generation sequencing were short read sequences. So you will fragment your DNA to short fragments and sequence millions of molecules in parallel, and then you align it back into that. And you all are familiar with the short read sequences. And the technologies they were using for short read sequencing were either sequencing by synthesis or sequencing by ligation. You remember I mentioned like first generation sequencing, they developed this sequencing by ligation, but the real use of that technology came only in the second generation of sequencing. Okay. Then came the third generation of sequences where people were using long read sequencing technology. So instead of using short fragments, they started sequencing bigger molecule, and that you can do using either single molecule sequencing or something called as synthetic long read. So synthetic long reads are not something used anymore. So but then most of the we will be looking at other long read platform. I mentioned like there is another fourth generation of sequencers also there, but essentially they are not based on sequencers, but rather than they based on approaches. So like all the single cell studies or the spatial technology where they are using microfluidics and other means to make a novel library prep method. So it's essentially the new library prep method was driving the fourth generation sequencing technology. So which is single cell technology or spatial technology. So sequencers were not generated, the new sequencer did not come during this period, fourth generation, but new library prep methods came which allowed you to look at genome at single cell level. Okay, that was the fourth generation of sequencing. So let's look at second generation. So I know most of you are aware of all these terminologies, but I was not sure about what is the level of audience. But based on your introduction, it seems like you all know these things. But just to summarize quickly, so what is a read? Read is basically a inferred sequence of bases corresponding to a single DNA fragment. So, as I told you, like we are sequencing millions of DNA in parallel. So, each molecule, the sequence originating from each molecule is actually called as a read. Okay. Now, I have a question for if anybody could actually give a stab to it. So, my question is why I am using this word called as inferred.
Because first we amplify our targeted DNA.
No, that has nothing to do. I'm inferring. I'm not actually saying. So, in case of Sanger sequencing, I was super confident. I said that this is a sequence.
It is not exactly by, so it occurs by fluorescence or by ions or something like that. So the sequence is read as a analysis.
Yeah, I know what you're saying. Yeah.
The reader of a signal.
Yeah, essentially, there is a third party software or vendor software which is telling us that this is the base. In case of Sanger sequencing, we exactly know that this is the base there, okay? But in case of this, we are inferring, okay? And so that is why there's always an ambiguity, like whether this base is right or wrong, okay? So that is why that's why it's not called as a true sequence, but rather than it's called as an inferred sequence. And the quality of sequences are so good nowadays that this really we don't have to use that definition anymore because they are extremely accurate. But still, for a historical context, it's always good to have that definition as a part of our discussion. Now, what is actually
I have a question. So what do you mean by read is the typical sequence? It
Yeah.
could be 10 base sequence.
Yeah, it can be any base sequence, but it should be originating from one single molecule. So you are sequencing millions of molecules in parallel, and each molecule will give you one set of signals. So if you are sequencing say 150 base pair, so you will get 150 base pair reads or sequence from each of this million molecule. Now each of them are called as a read. Okay. So now other thing is library. So those who are in genomics, you are familiar with this term library. So let me ask you, when you think about library, what comes in your mind?
Many things that
Yeah.
whole set.
Collection. So collection, okay, that's the first thing. So what is the nature of collection? Like, is it same book of 1,000 copy or different book of what is actually collection means?
It's a mixture.
Mixture. So it's mixture. It can be one copy. It can be 5 copies. Some of the books may have 100 copies. Some of them will be rare manuscript and you are not supposed to touch it. All these things. Okay. So the library is actually collection of molecules or collection. So when you have a collection of DNA and you want to sequence them, then you add certain things at the end of it, and that actually that is important for making sequencing. And once the molecule is ready for sequencing, those molecules are called as libraries, and they have same origin or have different origin, they have different molecules, different sizes, everything. Okay. Now the third idea is called as coverage or depth. So this is a very important question when you are doing any NGS experiment. The first question or two most important questions that you should be asking: how many replicates I need, and what coverage I need. Coverage means how many times a base is covered during sequencing, and why you need to do that.
Accuracy. Accurately we can determine the sequence.
Yes. So one of the example I can tell you, like if we are, so suppose if I have to tell you that go from here to say rock garden and document everything that you see in during your way. If you go once and then come back, then what will happen? That you may miss something. But if you are going 30 times, the chances like you will cover all the landmarks and all the information. So the coverage is very important and depends upon how rare the event that you want to capture. You will have a deeper coverage you need. Okay. And definitely we also need to learn about platform and how you calculate coverage. Coverage is actually calculated through, okay, thank you. So coverage is calculated through the number of reads you want. You multiply that with what is the read length. So how long you are sequencing those ones, and then you divide that by genome size. So if you are looking at RNA-seq data, then instead of genome size, you are looking at transcriptome size. Okay. So genome size versus transcriptome size. Okay, that's.
Slide move नहीं होगा.
No, that's fine. Okay. So basically, you take the number of reads you have. You go for the what is the length of the reads. So suppose if you are doing a paired end reads, then you multiply this by 2 and then divide that with the genome size, and if it is a transcriptome, then divide that with transcriptome. Okay, that is. Okay. Now, sequencing can be done as a single end sequencing or a paired end sequencing. Okay. So single end sequencing means a molecule can land either side, like 5' end or 3' end, and sequencer will go from one end to other end. By paired end sequencing, so sequencing will be done from one end to other end and then flip and then sequence from this end to this end. The idea is same what I was mentioning about our trip to rock garden. If you go once and just stop there and then come back, you never know that if you are seeing a difference in the basis, is it real or is it a error during sequencing. Now, when it comes to error, one of the main feature of error is errors are always randomly distributed. So same error will not happen same place, same time twice. It's always randomly distributed. So if you actually sequence same molecule twice from one end to other end and you see the same changes happening in the base, then you can ascertain that those changes are real. But if not, then then you know that there is some issue, and then you can correct it for those, and there are ways to do that mathematics. Okay. So now, when I started as a postdoc, that time sequencing was very expensive thing to do. I'm talking about $100 genome, $350 genome and all. But when I was doing, it was like if I have to sequence a genome, it will cost me around $10,000 to $12,000. It was 15 years back when I was a postdoc. So, and then paired end sequencing was like further expensive. But nowadays, the paired end sequencing and single end sequencing costs are almost same. So we are not offering any more single end sequencing because it's not worth offering single end sequencing. So most of those earlier consideration that whether you will choose a paired end sequencing or single end sequencing was purely based on necessity of the application. Some of the application where you need extremely correct reads, you will be actually using paired end, otherwise you will use single end because the cost was so much different. But nowadays, since the paired end sequencing costs are almost, or some cases actually it is cheaper than single end, so essentially we are using paired end sequencing for all of our applications at YCP. Okay. Now, let's talk a little bit more about libraries, the kind of libraries that we make. So libraries can be directional libraries, it can be non-directional libraries, it can be targeted capture libraries or amplicon libraries. So the directional libraries where you maintain the direction of the molecule, and where you need such an application. Can somebody guess where we need where we need to preserve the direction of a molecule? Anybody? Probably not plasmid. You are not going to sequence whole plasmid, no. Where do you think you need which molecule has a directionality in the genomics? Yes, maybe, but then where else? RNA is where you have the directionality. Okay, but again, there's another issue there with the RNA. You can always align RNA to a sequence, and then you can still find the directionality. Then why you need a directional library? No, it will, but then from a practical standpoint, if you can align your transcriptome back into the transcriptome database, you can still align it back, and then you say, okay, this is the thing, and then you can quantify whatever you want to do. Nowhere actually you read, in that case you don't need a direction, you don't need to know the directionality of it. Okay, but anyway, so let me actually give you some insight like why we need some of those directional libraries. So there are transcription, the way we think, especially in mammals, vertebrates, it's not that simple. There are a lot of transcription happens within a gene or from a So like a lot of genes are actually transcribed from the same genomic location in either direction. So there are divergent transcriptions are there, there are actually convergent transcriptions are there are transcript originating within a transcripts are there, the start sites are actually different from the same gene. So all these places are there where you can use the information related if you preserve the direction, because bioinformatics won't give you the directionality in real terms. So if you can preserve your direction, that means you can make more sense out of those mechanistic things. So for a normal bulk RNA-seq where you are just looking for differential gene expression, directionality is not that important. But if you are looking more into mechanistic details, you are more looking into those genes which are transcribed from a single loci or non-coding transcription. Some of them actually originate within the intronic regions or within the gene, and microRNA. All those areas actually directionality helps you to understand that data a little more. Okay, so you make directional libraries, and most of the cases directional libraries are made for RNA. Now targeted capture, a lot of you are working on that, exome libraries, or you can have a custom panel to capture it. Then you can also amplify PCR amplify regions, and then you can sequence them using NGS using amplicon libraries. Okay. So one of the key idea of sequencing, and you will be seeing some of this when today afternoon when Francis will be going to talk to you about FastQC, that the Q scores. Okay, so sequencing quality is given in terms of Q scores, and Q score stands for 10 to the log 10 E, where E is the estimated probability of incorrect base. Basically, you are looking at what is the probability that a base called by your base caller is incorrect, and if it is Q10, which means every one base called from out of 10 will be wrong, which is like really we are sequencing a million base, that means you are almost 100,000 bases, 1 lakh base will be wrong. Okay, Q20 means like 1 in 100, which is around 99% accuracy, and Q30 means 1 in 10,000. So the new sequencers, so Illumina sequencer operates at a level of Q30, that means 1 in 1000 base error. There is a new sequencer called as AVITI. The sequencing quality is in Q40, which is 1 in 10,000, and they also came up with a new chemistry which can give you Q50 read value, which is 1 in 100,000 base error. So which is extremely accurate sequencing done by these platforms. Okay. So NGS platforms, when you think about platforms, there are various platforms available and they differ in chemistries. And then one of the key characters of NGS or next generation sequencing, that they can sequence millions of clonally amplified molecules in a massively parallel manner. So this is the difference between Sanger sequencing or first generation sequencing. So two things, that you should be able to sequence millions of molecules and in parallel, which means every single molecule is sequenced along with others. Okay, and instead of doing this gene by gene, you can actually do everything together. So what actually really differs in case of various sequences is that how you do clonal amplification and how do you reaction, like how you measure the output of that and how do you do the clonal amplification. Now, can you tell me why we need to do clonal amplification? Why we need to amplify that signal? Why we need to do that?
Accurately, we can determine the sequence if there are more number of molecules, more number of copies of that molecule.
Why?
More number of times it is read. More number of times the thing is read again and again.
No. So this is the clonal amplification. Clonal amplification actually is defined by in a sequencer you load one molecule or say one spot you have, and on that spot itself it amplifies and it actually creates a bundle of fragments there.
So all the signals should be so for base accuracy.
Signal
It's very
amplification.
important that the fragments is
Signal amplification.
Yeah, so signal amplification. So signal to you want to achieve a high signal to noise ratio. So here is my question. Then why don't we use a very bright floor? So instead of amplifying signals from a multiple floors in a bundle, and get a very high signal to noise ratio, just put a very bright fluor, which is really bright in a dark background. That means you will be able to get it.
Sometimes wrong bases are called. So
No, I'm fine with that. I'm sequencing millions of molecules. I'm fine with like 1 in 100,000 molecules being called, 1 in 100,000 bases being called wrong. I'm fine with that.
Some genes are very low in copy number. Some genes have high copy
No,
number.
it has nothing to do. You are not talking about gene here. We are talking about fragment. Each fragment we are amplifying. We are keeping a bundle. And I agree with your idea that we are actually doing that to increase the signal, and then that way we are actually increasing signal to noise ratio. So we are reducing the noise and we are increasing the signal. So now it's like night sky, you are actually seeing a brighter star. Okay. So my question is, rather than doing it like, so in this case you are using a dim fluor and you are using multiple dim fluor and you are aggregating all the signals together to create a enough bright signal, and then you are capturing it. But instead of that, why don't we actually put one single bright fluor and say like, okay, I'm done with this, I don't want to do clonal amplification.
I just want to guess. Do we have a fluorochrome like that, number one? And number two is the spectra varies, overlapping with the other colors maybe.
We have, believe me, we have a fluor like that. Okay, we have a fluor, and if not, then somebody can make a fluor for that.
Sensitivity.
Okay, hold on to that thought. Okay, sensitivity. Then anybody else? Okay, so let me actually
So
give.
signal will be too bright that we may have problem in reading that signal.
Yeah, that is a problem. So there are two problems there. If you have, so how this technology works actually? There's a high-speed camera. It's actually taking pictures. Okay, so if you have a very bright fluor, then what will happen? The two molecules, they are actually near to each other, the signals will merge. Then the camera won't be able to resolve it, which means like you won't be able to sequence millions of molecules. Rather than now you have to reduce the number of molecules and some way, and you have to space it enough, okay, so that those signals can be. But there is another bigger challenge for also. This is a technical challenge, but there is another financial challenge. When you actually employ a brighter fluor, the sequencing reaction will become more expensive. So it's not that we don't have brighter fluor, we have brighter fluors, but the cost of sequencing will be dramatically high. So that means that both the things, like you want to control over the cost of sequencing reaction, at the same time you want to make it as dense as possible. Like I was talking about NovoSeq X Plus platform, in single flow cell, this is such a small flow cell, and it can sequence up to 25 billion reads. That means it's such a dense environment that the molecule to molecule you don't have any much space left, and if you have a bright fluor, then it will blind everything, and then you won't be able to resolve it at all. Okay, so the most important thing, right, that you cannot resolve it, and then cost is actually very high when you are using bright. So we clonally amplify. Okay, now for the pyro sequencing you use emulsion PCR. Same for the the sequence mobilization, also use emulsion PCR, and then ion torrent you also use emulsion PCR. While for Illumina based sequencing you use cluster generation, and we will be seeing this process a little more detail. And the output is captured. For the pyro sequencing we will be using luminescence, while for solid we are using fluorescence. Ion torrent we are using pH change, and then for the fluorescence based imaging we are doing for the. Okay, okay. Well, yeah, 10 minutes. Okay, so let's start with some short read sequencing platforms. The first one what we are going to look at is ion torrent. Okay, so ion torrent actually senses pH change. Now how it is, what it does, you have a DNA, and then you have a primer. Actually for the sequencing primer you add the sequencing primer, and sequencing primer will go and align to this. Okay, after that you will flow your first base. So you are here, actually you are adding one nucleotide at a time. So you will add your first base, and then you try to measure that if anything happens in the pH. So if there is a complementary base pairing, then there will be a release of an H plus ion, and that will change the pH of this micro well. And that change is actually sensed by a pH sensing layer, And the moment that pH change is there, it will register that, oh, once you flowed, if the flow, this particular base was flowed, you have that pH change, and then you register that as that base. So like in this case, as soon as T, if A comes, nothing will happen. There's no chemistry. Kuch kuch hota hai type. If you don't have kuch kuch hota hai, then you will not see that. So the moment T comes, then you say, okay, kuch kuch hota hai, and then you sense that kuch kuch hota hai in the pH layer, and then you will. So this is through sensing that H change in the ionic concentration, H plus ion concentration in that local environment, and that will give you, and that is how you are doing the sequencing. Okay.
There will be always change in one edge, right? So how will it differentiate between the bases? So when there is a complementary reaction, though there will
Yeah,
always
so
be the same change in pH, or there will be variation?
no, there is the same change. So what happens usually that you flow first base. So that is what I was saying, that you suppose in this case the first base is A. Okay, so you say the first base came inside the flow where is C. C, nothing will happen because there is no base complementarity. So and then you wash off the C, everything from the well. Then you put second one, then the third one. So whenever the real complementary base comes, the H plus ion the change will happen, that will be sensed. Then you wash off that reaction. Now the well is back to his original state, but at that point you have the first base already reverse complement. Now you are looking for the second base. So now you are looking for G to G. So like that it goes and it does in
Depends upon we know the flow of the bases what we are throwing at point.
yes,
In relation to the flow of
the
which is
nucleotide.
coming in.
Yeah, the basis which is coming in. This is a very simplistic view of ion torrent reaction. There is certain primer based jugglery is also been done in this, but the basic idea is this: like your base comes in, if it is actually a complementary to to the base that is there, it will release a H plus ion, and that H plus ion will be sensed by this pH sensing layer, and that.
But the only problem would be when there is a string of same nucleotides.
Yeah.
Homopolymer repeats, because there are a lot of string of A's or string of G's. Then to differentiate that, how much of that is because of 4 or 5 or 6 or 7 becomes good. And so this kind of chemistry is very poor for regions where there are homopolymers.
But unfortunately or fortunately, this is one of the most widely used platform for clinical application. A lot of faster. I think, yeah, clinical setup. You see a lot of ion torrents working in clinical setup. Okay. The other platform is called as nanostring. Okay, so it's encounter platform. This is basically this is a counting platform. So you are not sequencing here. You are just counting. And what are you doing here? So suppose this is your so it has each gene is actually characterized by two sets of probes. One is called as capture probe, and another one is called as reporter probe. And reporter probe will have a set of fluorescent barcode. At each of the reporter probe will have a series of reporter barcode defining each of those genes. When you hybridize, then the capture probe and the reporter probe will go and hybridize to the gene. So now the one side is for pulling it out, and second side is for reporting. So unless you have both the sides on the same molecule, you will not be able to do anything. So once the right gene is there, then that means the capture probe and the reporter probe will go and hybridize to the gene of interest, and now you wash off anything that is not hybridized, and that means you are left with a series of hybridized molecule with a distinct combinatorial fluorescent makeup, and the reader will go and read and just count like how many of them are there. That's it. It will just count like, okay, this particular combination of fluorophores, this many number, this, and then that you just convert that into name of a gene, and this is how and then you end up having a data something like this. So here you are not sequencing, but rather than what you are doing, you are just counting. You are just counting, through hybridization. And
So it's about the gene expression.
yeah, it is mostly used for gene expression analysis. So again, this is one of the platform that has a lot of traction in clinical setup, because you can make small panels like 60 gene, 80 gene panel, and instead of doing RNA-seq and then doing all this complex analysis, you can quickly get the results out of those clinical samples. If there is an assay you can develop on this. A lot of people actually love to do this for cancer and other area, but this is again because of the simplicity. And the only issue with this platform is an initial cost of setting up this, because those synthesis for this probe is expensive. So once you synthesized it, it will be for hundreds and thousands of reaction. That means per reaction cost is low. But if you have only few samples, say I have only 10 samples, probably I will not go for this kind of panel. But if you are going to use a panel for repeatedly for your clinical application or in your lab, say like your rest of your 5 years you are going to do thousands of samples, then this is the cheapest way to do a gene expression analysis, and you don't need any bioinformatics support to do so. Okay.
Besides cancer panels, like what other applications this encounters
No,
besides cancer?
no, everywhere, wherever you have a gene expression. Gene expression. You- yeah, and like best example is infections. So like you have a RSV panel, or you have a respiratory virus panel, 200 virus panel you have. You just create a set of like probes and reporter gene for each of them and run it. Like within a minute you will be able to quantify. And the good thing about it, it's sensitivity. Even for single virus level, sensitivity it can give you with those. But again, the main issue with encounters are that unless you have enough number of sample to analyze, it's not worth, because of the initial cost of generating the probes and reporters are actually high. But if you have enough number of samples and you are going to use that for over years, then I think this is a great platform.
What is the advantage of this over droplet digital PCR?
Droplet digital PCR, so two things. One, droplet digital PCR is still very low throughput. You cannot, I mean, most of the platforms cannot go, I believe Coagility is the only one which can go up to 12 floors. But most of them are between 4 to 5 floors. So that means you cannot analyze more than 5 genes in a droplet digital PCR. And it's expensive. The reagents are expensive. The Tachman probes are expensive. The CyberGreen is expensive. Everything is expensive there. So that is what I was saying. If you have only few samples, say like 10 samples, probably digital droplet PCRs can work. But if you have enough number of samples, and then if you're looking for 60 or 80 or 100 genes, then the encounters are the one. For larger number of genes, encounters are, droplet PCR good for like fewer genes. And if you have a larger number of sample, again encounters is that one.
So reported dyes of multiple fluorescence.
Yeah, they actually use what they call, what is that? Quantum dots.
Quantum.
Yeah, so when I was actually presenting, I was thinking of that term, and I was not getting it. So thank you for asking me. So they use quantum dots, and then they actually create a combination, and then that is what they read.
So each reported dye will have for each gene, or they will give
Yeah.
a combination of.
So yeah, they give a combination of quantum dots, and that combination is unique. And the encounter, the instrument reads that combination and deconvolute back into the gene of interest.
This encounter technology, is it, IVD approved for breast cancer?
I'm not sure about the IVD approval, but in US there is a way we actually called as LDT, lab developed test, and which can be, so basically the difference between IVD and LDT is that LDTs are usually done by individual labs. So like my lab, I can develop an LDT and I can validate those LDT based on CLIA cap regulation guidelines, and then I can offer that service to anybody I want. But that cannot be used outside my lab, because it is a protocol, a assay that is validated just for my lab. But IVD is something that you do a large test, you file it with FDA. FDA will give you approval, and that means once you have an IVD approved, that means anybody can use that technology by paying a fees to you. So I'm not sure about IVD part, but I know that a lot of labs are actually using this under LDT licenses. Okay. Now, just before, okay, I think I will stop here probably, and then we'll have tea. And if we are left with like 10, if you come for last 15 minutes, I will use probably, or you can actually take it from there after tea. No, you start, and I will continue next like day or.
So we're going to have a break now?
Yeah, they will have a tea break for 15 minutes, and then you can set your things up, and then we can go.